Okay, good morning. Our first team talk today is presented by Agnes Bodor, Tim Jarsky, Stefan Michals, Brian Lee, and Forrest Coleman. In this talk, they'll present the Institute plans to characterize the diversity of synapse types, including the full dynamic properties of synaptic physiology. Over to Stefan.
Morning. So we are interested in linking, getting to function. What do we mean by function? In the case of an animal, for example, the function is linked to behavior. In the case of a visual scene, what is needed to be done are local computations, for example, detecting local features, taking this type of features, putting them together into an object and identifying them. However, what we want to do is to link the structure towards this function. The structure is enormous. When we talk about the cortex of a mouse that has about 5 million neurons and tens of billions of synapses. In this movie, we can actually see that even on a small piece of cortex, we have to zoom in quite a while in order to be able to get to these synapses. So how can we make order among an element of the orders of tens of trillions? What we have to do is to think of principles of organization. So we start by first characterizing the elements which are going to be of spatial connectivity. In here, what we mean is different regions in the brain, how they connect to each other. After that, we want to go into slightly more details in which not only particular regions connect to each other, but each of these regions has a high diversity of cell types, and there aren't a unique set of cells which are projecting from one place to the other. At the same time, there are good principles of organization. For example, we know that the cortical structure is not totally random. It's not tens of trillions of random numbers, but there is a high level of symmetry, and there are a good set of rules which we can discover. For example, only on the order of 10,000 genes contribute to formation of the cortex. And on the lowest scale, there is individual connectivity, which is going to be the central theme of the next talk. What do we do from the perspective of modeling? We have a set of measurements of individual connectivity, and from these what we want to do is to generate a set of abstract rules in which we generate a set of cell type connectivity, and later on, we can either simulate these directly, and I would recommend you later today to visit Nick's poster 28, to see of a method in which we can go on and directly from this set of abstract rules run simulations. But most of the time, what people do is they generate a network instantiation. In order to do that, we need to have a set of assumptions which are coming in from this set of rules. So, for example, we can say, oh, if we have a general connection probability between two cell types, they can be randomly connected. But at the same time, that doesn't typically work very well. We need to use some more interesting functional assumptions. For example, that they do an interesting function at the level of individual cells to connect them. What we use modeling is for two elements, to link structure to function and to think of ways to prioritize measurements. To link structure to function, we don't start at zero when we characterize cell type specificity. There is a small microcircuit for three types of inhibitory neurons, which has been characterized in terms of parvalbumin, somatostatin, and VIP neurons. What we do is and I would recommend for a lot of more details visiting poster Jung-Hong Lee's poster 29 later today, construct a set of, in this case, for example, 13 cortical columns, and we implement local and long-range circuitry, which is cell type defined. And what is very interesting is that for different inputs to different inhibitory neurons, we find specific functions. For example, pyramidal inputs to parvalbumin neurons are super important for border detections. Pyramidal input to somatostatin neurons play an essential role on the competition between multiple objects, and top-down inputs coming into the VIP neurons have the capacity to do gain modulation. So we've seen that we have the capacity to map these type of cell identities and their specific connectivity to specific functions. However, we just barely scratched the surface on the cell type diversity. You've heard yesterday that at least even if we start in V1, there are at least 42 cell types which are characterized from the perspective of the transcriptomic, and in here we use the small minority of them. So what we are interested in is being able to get considerably more data, but the part which it is what is the most important data to take, and for that we used sensitivity analysis. What does it mean is that we modify a little bit the parameters in the network and try to see which are some of the most important parameters for the output of the network. And we've seen that there are many types of measurements which are not always measured, which play an important role. For example, multi-spike short-term synaptic depression is very important beyond the synaptic weight and the properties. So the fact that we, as modelers, and there are many other modelers which we want it all, always. But at the same time, we need to think as to what can be prioritized. And in this case, we want a set of rules which correspond to synapses.
Thank you, Stefan. So I'm the first to tell you about a multi-scale experimental effort ongoing at the Allen Institute to characterize and understand the properties of the connections between neurons. So I'm part of a growing team that's using multi-patching to look at the connection properties or to dissect the properties of local circuits. And so today I'm going to tell you a little bit about the type of data we get when we study these local circuits. I'm going to tell you about why we use the approach we're using to dissect local circuits and some of the trade-offs. And then I'm going to finish with a summary of some of the automation we're using to overcome the limitations of the approach. So in the type of experiments we perform when doing in multi-patching experiments, we're recording from many neurons at once. And we use this experimental approach because we can resolve tiny and quick signals. And an example of which is shown here on the left. So in this case, there's a recording from two neurons shown, one here and one here. And in the first neuron, we're injecting a short current injection that induces an action potential in the injected neuron that's shown in truncated form here. In the uninjected neuron, we see a corresponding chemical synaptic transmission event. And in this case, we know it's an inhibitory event because it's blocked by an inhibitory synaptic transmission antagonist, SR95531. So we can then look at the properties of this response, and extract features. And we can use these features to characterize different synapse types. At the same time, or in the same experiments, in addition to studying chemical synaptic transmission, we can also look at the electrical junctions that occur between some neurons. So instead of the short current injection used to elicit an action potential, we now use a smaller amplitude but longer duration current injection, sufficiently long that the voltage response in the injected neuron reaches a steady state. And now we can look in the uninjected neuron for a scaled, smaller version of the voltage response, indicating the presence of electrical coupling. So unlike chemical synaptic transmission, electrical coupling is always bidirectional. So now, in the previously uninjected neuron, if we inject current, we can now observe a nearly identical voltage response in the uninjected neuron. As before, we can extract features of these responses to characterize the electrical junctions formed between these neurogliaform cells. So this is an example of the high-fidelity data we can obtain with multi-patching, and it's really unique to this approach. We can then pool sets of experiments to look at the connectivity as a function of distance. And so I'm showing you the synaptic connectivity as a function of distance, in this case for two connection types. Those formed between neurogliaform cells, or NDNF positive cells, and those formed between parvalbumin positive cells. In both cases, these are connections studied in the primary visual cortex, and these cells are found in layer 1, and here we focused on cells near layer in and near layer 4. And what you can see from this distribution is that the neurogliaform cells have a much broader range of connectivity. We can also look at the property of the electrical coupling in the neurogliaform cells as a function of distance. And what you can see is that the strength of the electrical coupling falls off around 150 microns, consistent with the fact that these gap junctions are known to be dendrogingitic. So this is the type of high-fidelity data we obtain with multi-patching to understand the connections formed in local circuits. Another reason we like the multi-patching approach is that there's this steep nonlinear relationship between the number of simultaneous recordings and the number of probed connections. So we're highly motivated to increase the number of simultaneous recordings because it allows us to probe proportionally much more connections with each recording that we add. Unfortunately, there are many practical limitations that prevent us from increasing this number. And one way to quantify those practical limitations is shown here on the right, where we repeated multiple quadruple recordings attempts and quantified our success rate. And what you can see is that with multiple quadruple recordings, we more frequently obtain double and triple recordings. And this success distribution corresponds to a mean individual patch success rate of around 50 to 60%. So I think there are a couple of approaches you can take to overcome this, and I'm going to describe one that our team is working on, and that is to make the cost of a failure much low by automating multi-patch experiments, so that if there is a failure, making another attempt the cost is so low that it's nearly insignificant, or drive it as close to that as possible. So I'm going to show two quick movies where we've automated a couple aspects of the multi-patching experimental procedure. Shown here on the left are examples of the coordinated and parallelized movements of multiple manipulators. I'm showing one where we're moving the manipulators in parallel down to the tissue, and a second where we're coordinating the movement of manipulators into the field of view of the microscope. On the right, I'm showing the automation of pressure regulation. So in multi-patching experiments, we use pressure to form a seal between the electrode and the cell. And then we also use pressure to rupture the membrane inside the electrode. This allows us to form a closed electrical circuit, which allows us to record these small voltage and current events that these cells exhibit. And so what we're showing here is simply as we place the pipette on the cell, we switch from a positive approach pressure to a negative pressure. In the seal, you can begin to see the electrical resistance rise, and then once it rises sufficiently, or we observe dimple formation, the sealing algorithm is initiated. So this will come here in a moment when you see this button go red, and seal formation begins. You can see the electrical resistance rise, and now we have a high resistance seal between the pipette and the cell. And then we can begin the break-in algorithm that ruptures the membrane and now gives us electrical access to the inside of the neuron. So these are just two examples of the automation efforts that are ongoing at the Allen Institute. So I've told you about our multi-patching approach to characterize local circuits. And now Brian Lee's going to introduce our efforts to understand the properties of the long-range connections.
Thank you, Tim. So multi-patching recordings provide us with a wealth of information. However, the throughput is quite low, and as Tim mentioned, without automation can be quite laborious. So we're using a method, Optogenetics, that allows us to complete this matrix in a more efficient, albeit less informative manner. So with Optogenetics, we will focus on the LGN to V1 projection. And with this projection, we can measure particular parameters such as STP and receptor composition within specific cell types of V1. So when we assess local networks, as Tim described, it's often done in coronal or sagittal slices, which range from 250 to 350 microns thick, and can only examine connections within that fixed plane. So when we examine the long-range projections as I described from the LGN to V1, the soma is often severed, which renders these multi-patch recordings with this pathway impossible. So with the advent of optogenetics, this allows us to study the long-range circuits despite the lack of a soma. So therefore, we have taken this approach to dissect neural circuits, essentially using blue light to stimulate neurons. And what we plan on doing, or what we are doing, is we package the channel rhodopsin protein into a virus expressing a yellow fluorescent tag and express it in the LGN. And then this will allow us to take a coronal slice that contains light activatable processes within V1. So now that we've have the LGN to V1 pathway isolated, we can take this a step further and inject the channel rhodopsin into the LGN of a specific Cre line. This allows us to take a very refined approach to evaluate a specific pathway onto a specific cell type. And for the purpose of my study, I've used this with 3 different layer 4 Cre lines, the ROR-B, the SCN1A TG3, and the NR5A. And what we have here are some low magnification images of V1, and this top panel, the top panels show the LGN YFP processes within layer 4. And then this middle panel is another low magnification, just showing the TD expression of the particular cell types within layer 4. And then down here is a 40x image showing that indeed that the cells that I patched onto are the TD expressing neurons. So once we have this preparation, we can conduct a multitude of whole cell electrophysiological experiments. And one experiment is to examine short-term plasticity. So with this, we can take light and stimulate take a pathway-specific approach to apply trains of stimuli onto LGN fibers. And we will do this with the 3 aforementioned Cre lines. And so here we have the traces from each individual line, and here are the indications of the light pulses that are applied to the slice. And we can measure the light-evoked response and the degree of depression, as well as recovery test pulses at varying times. And we can plot these data points and quantify the curves, especially the recovery test pulse. And we can quantify the curve and delineate any potential differences in the LGN presynaptic input onto specific cell types. And what we show here is that the SCN1A and the NR5 seem to be similar, and the ROR presynaptic recovery from depression appears to be slightly different. However, I think we need to include more cells to try to make that claim. So complementary to the presynaptic side, we have the postsynaptic side. And since LGN afferents are largely glutamatergic, the postsynaptic membrane has 2 main ionotropic receptors, the AMPA receptor and the NMDA receptor. And we have a method to identify those specific receptors using pharmacology. And here, again, we have a low magnification image, except for this time I have both fluorescent channels merged onto one. We have the ROR, we have the SCN1A and the NR5, and you can see the TD expression for each of the within layer 4, and then you can see a faint YFP image overlaid on top of that, and then the 4x images of the particular cells patched. And so what we do is we voltage clamp the neuron at -70 and +50, and we apply voltage clamp the neuron at -70 and +50, and then we apply a 3 millisecond pulse. And at -70, you can see that the large majority of the current is mediated by the AMPA receptor. And as we depolarize the cell to +50, we alleviate the magnesium ion, and now the current is mediated by AMPA and NMDA. And to isolate the individual receptor components, we can apply NBQX, and that blocks the AMPA component, leaving a pure NMDA component. And then we can calculate the ratio of the NMDA to AMPA ratio, and that is quantified here on the left. And as you can see that within these 3 afferents onto these or the LGN afferents onto these three specific sites, there appears to be no difference within the receptor composition. And then with that, Forrest is going to go on the anatomical methods.
Thanks, Brian. So up to this point, Brian and Tim were telling you about the Institute's work to characterize cell type specific connectivity in terms of functional assays, where they're recording the response of one neuron over time as one or more other neurons fires action potentials. Now, this, of course, is not the only way that you can measure connectivity in the brain. You could also do it structurally or anatomically, because, of course, underlying each of these functional events is a set of synapses, which are the physical basis of this phenomenon. And anatomical methods have some advantages, maybe most importantly that it's easier to imagine how to really make anatomical measurements at a much larger scale than physiological ones. And this movie here, which you've already seen in Stefan's introduction, kind of illustrates that point. This is an image of an ultra-thin serial section. It covered about all the layers of cortex, about 1 millimeter tall, 500 to 800 microns wide. The cyan shows DAPI in the cell nuclei. As we zoom in here, the red shows a postsynaptic marker of excitatory synapses. Green is a marker of presynaptic zones, and blue is GABAergic processes. And you can see, zoom down here to an individual synapse. And I think that you can appreciate just how many synapses are packed into even a single ultra-thin section. There's on order tens of thousands of synapses that you can find in one section. As you start imaging many sections, the numbers really grow quite astoundingly. Now, of course, a purely anatomical method has some obvious disadvantages. Maybe most particularly, you're missing two critical pieces of information. The first is, well, what is the functional significance of this kind of structural measurement? We are ultimately interested in understanding the brain in terms of functional terms. We want to know how each of these different neurons influences the dynamics of those that it's connected to. And so what is this relationship between the structure and the function? And the second, of course, is, well, what are the cell types involved in this synapse that you can see here? What type of neuron is the presynaptic neuron? What type of neuron is the postsynaptic neuron? Because we're not interested just in counting synapses, but rather understanding this complex cell type specific connectivity that Stefan described at the beginning. Now, although it's not immediately obvious what those are, there are very good reasons to believe that this information is encoded in the brain. From the number of synapses, to the size of the postsynaptic density, to the morphology of the dendrites and axons, to the nature of the complex molecular signaling networks that exist in synapses and in dendrites and in cell bodies, there are a myriad of ways in which biology encodes information in the structure of the brain. In fact, I'd argue that at some level, all functional signals and everything has to be encoded at some level structurally, because every phenomenon has a physical basis. So the question isn't whether or not the information is encoded. The question is, how is it encoded, and what do we have to do to read it? So in order to be able to answer that question, what we need are a set of Rosetta Stone experiments where we measure both the structure and the function, or both the structure and the cell type, in order to learn how these two pieces of information relate. So one way that you might make the functional version of that experiment is to do exactly the kind of experiment that Tim was talking about. Here, for example, is a recording that Brian took, in which he's recorded carefully characterized the electrical and chemical synapses between these two neurons that he's patched onto. But we can also fill those neurons with two distinct dyes, and then we can attempt to take that same exact slice and attempt to reconstruct, using array tomography, those precise neurons and find the anatomical connections that underlie that functional synapse. And we're working towards doing these kinds of experiments, and we hope that by building up a large set of examples of experiments such as this, we can build a larger and more complete and more complex dictionary that allows us to translate the vast amounts of anatomical data that we can collect into the functional language that the brain speaks. And so with that, I'm going to now let Agnes talk about the second version of that Rosetta Stone experiment to understand how structure and cell type relate.
Okay, so Tim and Brian just showed you how to investigate connectivity in the brain with physiological method. in slices, and Forrest showed how we intend to relate the anatomy to the physiology in those slices. In order to calibrate these measurements to the circuits found in vivo, we need to identify these synapses in a bigger volume of tissue, which is about a cubic millimeter, and we need that large because we would like to have the complete W alpha beta including in it. And to do this, we have two different strategies. One includes morphological methods, and another includes genetical tools. So now I'm introducing the first one, which includes the anatomy. For that, we need electron microscope, and also what we need to do, similarly how Forrest showed, we need to slice the synapses, and average cortical synapse take about 20 to 60 40 nanometer thick serial section. You can see them in this grid. And later, when we doing the cubic millimeter experiment, we need to cut 25,000 serial section. So here you can see the synapse from the thalamus to the V1, what Brian already introduced. This is his view. And this is how I can see the same type of synapse in a electron microscopic tint section. What you can see here, in the middle, it's a synaptic terminal from the thalamus. I know it's from the thalamus because you can see it's darker, it's labeled. And this synaptic terminal contains the synaptic vesicles. And you can see that this terminal contact two dendritic spine, which is the most common target in the neocortex. And you can also see the presynaptic side, the density, and also in the postsynaptic side. The presynaptic side contains the docking apparatus and the proteins helping the release of the transmitter, and the postsynaptic side contains the receptors. So when I looking in the electron microscope, I'm seeing an image like this. This is a neuropil, contains several synapses, so I can point out at least 5 of them. And you already can recognize that there are some morphological differences. For example, this one is much larger than this one. The target element here is a dendrite, and it's a spine here, and the active zone size also different. But I still cannot tell you which cell type forming these synapses. So to overcome these difficulties, what I did, I used immunohistochemistry method to label specific cell types. For example, what I'm showing here is a layer 4 cell type, a layer 6 cell type, and the thalamic input. And I made serial sections from them. And based on that serial section, I reconstructed them in 3D. So the purple is the thalamic axon. And here are some examples from the layer 4 in red and the layer 6 from, in blue. And so I can make very careful measurements. For example, I can measure the active zone size. I can see how many synapses formed by a single synaptic terminal, or I can define the postsynaptic element, and so on. So after I reconstruct hundreds of these synaptic terminals, I have a little data set, and if I compare the different cell type specific synapses to each other, based on my current data set, I can differentiate these 3 type of synapses with a 75% certainty if I use all the data together. But sometimes we cannot differentiate cell types just based on morphology. So this is when we can take an advantage of the Institute large-scale program to build genetical tools. And one of these tools include a genetical tag which are directly visible in EM in the electron microscope. Here I show a cell which is labeled, the whole cytoplasm. So here it's the soma, and we can see the presynaptic side, the spine of the same cell, and also the synaptic terminals. And we are hoping that combining these two methods, we can identify a substantial amount of alpha and beta in the volume of cortical tissue, which is enough big to contain complete microcircuits.
So what we have heard are physiological methods which allow us to have a very detailed view on a lot of functional properties which we are interested in, about anatomical methods which are capable to do that on a much larger scale, and combined methods which give us the possibility of linking all this entire data set together on a much larger scale. But even with all these methods put together, the huge scale of the entirety of the W alpha beta is probably not going to be able to be tackled fully experimentally. What we need to do is to be able to learn to generalize from the data which we have. And for that, we need to have an additional set of assumptions. From this perspective, there are some very good methods in the field. For example, morphology puts a nice set of restrictions as to which are the types of neurons which can connect to each other. This has been apparent in the very recent paper from the Blue Brain Project, which take a good advantage of this morphological knowledge. It is possible, if we have this type of data, an anatomical and physiological measurement of the connectivity together with this, to be able to learn more of the structure, how to be able to put together the morphology. On top of that, we have the capacity to be able to define further the cell types from what we currently have. A second approach is going to be integrating the fantastic transcriptomic data which we have. We know that all of these elements are going to be present somewhere in the genome. There have to be pre- and postsynaptic markers which are present. If we have a large number of experiments and with a good knowledge of the transcriptional profile of these cell types, it might be possible to be able to learn to generalize to some of the unmeasured elements from the transcriptomic element. And on a final level, there is a functional element as well. Not only that this is a brain which is a mature brain, which has actually spent time in context, looking at natural scenes, understanding and interacting with the environment. So on top of the structure which we learn from the morphology and from the genetics, there is going to be a functional level on top, and we want to be able to learn, to be able to link the cellular responses in a natural environment to the type of structure which they are forming. In conclusion, we started with a very short motivation. Why do we want to have this type of mesoscopic cell type specific description of the connectivity, and focused on 3 methods to describe the cell type specific connectivity. A physiological method, several physiological methods based on multipatch and 1P optogenetics, which provide data which are directly usable in the models, but they are on a smaller scale. Anatomical models which have the potential to be able to build up more of this scale, and the computational models which allow us to, I would say, get informed guesses for the elements which haven't been measured. Thanks.
Okay, thank you, team. We have some time for questions now, so if you have a question, please raise your hand and we'll come to you with a microphone. Sean.
Very nice talk. So my question is about the extent to which you think the synaptic properties measured in vitro will translate to in vivo properties.
We only have to adjust the calcium level.
What about neuropeptides?
Okay, sorry.
about the neuropeptide milieu?
Not to continue with the joke, but we want to be able, in the end, what we want to do is to be able to find this property in vivo, and the elements which are, we know that there are several elements which are going to be different, and that is, at least from a computational perspective, something which can be tackled by having the capacity to search for functions. We know several functions which are going to be present in vivo. I would really love if we can convince Tim to do octopatch in vivo, but I don't think that that is a feasible technique. What we probably want to do is to be able to learn from this data and learn a way to map them to generalize.
Question over here. Thanks for the great talk. My question is, what are you not doing? In your three categories, does everything fall under those umbrellas, or are there other major areas that you're not, or tools or approaches you're not using? So, for example, perturbation, that's not on? Oh. You all heard that, though. No. My question's about what you're not doing. Are there, you've got your three umbrellas. Does everything fall under that, or are there other major areas you're not tackling? For example, perturbation with drugs or with ionizing radiation or by genetic background. Are you all going to do this, or have you, are these separate approaches that you have decided not to use?
So currently I'm only analyzing the layer 4 input, and only in like animals which are, some are genetically modified because the label, but that's all. I'm not doing any functional comparison regarding the synaptic terminals, although it would be very interesting, but it's a very, very time-consuming method and very hard.
Go on, Forrest.
I mean, I'm speaking for the physiologist here, but I mean, I think one major thing that I don't know of plans to do immediately is examine how synaptic properties are modified by neuromodulation, which is obviously hugely important, but it's not on the in the immediate plans.
Or one interest of mine would be plasticity mechanisms in dark-reared animals. That's definitely an interest of mine, but I don't see any current plans to examine that.
Okay, next question, please.
How many different genetic labels can you visualize in a single preparation? You showed one picture of genetically tagged cells, but can you genetically tag multiple types of cells to try to pursue the cell type specific connectivity further?
Yeah, yes, so it's actually require more different type of animals. So currently the hard part in electron microscope, we have only one color, which is the diaminobenzidine. But if we are able to make animals where a given cell type express this tag, for example, in cytoplasm or the mitochondria or the vesicles or in other elements, so different elements in each cell type, we will be able to label probably several. And there are literature on this, so people are working on in similar models, and it's working.
Forrest, want to keep going?
Stefan, you mentioned that you are going to use models to determine which parameters you would like to know most urgently. So can you say some more about what these models are going to, are they going to model, orientation tuning, or can you say some more? How is this going to go?
This is an iterative approach, because that is one of the biggest problems. The current models which we looked at were at the level of firing rate responses in a set of balanced networks with particular predetermined topology, such that every time when we get more information from the experiments, we want to incorporate that, go back to the models, vary the parameters, some of which we know maybe a bit less, and some of them which we don't know, compute how much the changes in those parameters affect different functions, which at the moment we focus on firing rates, but there may be many other ways in which the coding is going to go. And then we are going to come back and just to say, oh, right, now the most important parameters, the next set of experiments are going to be where these are the elements. Currently, we are doing this at a very shallow level, but it is an iterative process which we intend to apply. We intend to work in tandem with experimentalists to iterate on this process continuously towards better functional descriptions.
Okay, thank you.
Okay, we have another question.
So how does this effort intersect with the plan to reconstruct all the processes and neurons and synapses in the cubic millimeter, which I think is one of the milestones on your milestone program?
So this is why we're analyzing the synapse, and we try to learn their morphology. So in a cubic millimeter, we barely can use immunohistochemistry because a lot of penetration and other issues. So probably we will depend on the morphology. So this is how it's related. I'm learning the morphology of different cell types, and when in a cubic millimeter, so we cannot differentiate them usually 100% sure. It's currently these 3 cell types we can differentiate based on the synapse at 75% certainty. But imaging in a cubic millimeter, we will have much more information. We will have a long axon with several synaptic terminals and other elements too. So we are hoping that the morphology will be one of our key tools to identify the cell types. And also we have this genetic tag, which is directly visible in EM level, and the penetration is not as big as a big problem as with the immunohistochemistry. We have much better penetration. And if we will have more than one animal, for example, some animal can express, as I mentioned, intracellular or extracellular or mitochondria. The tag, we will have probably few cell types also labeled.
Okay, thank you very much, team.